Cloning, once the realm of science fiction, has now emerged as a fascinating and controversial reality. The concept of creating genetically identical copies of living organisms has sparked heated debates within the scientific community, governments, and society as a whole. While cloning has shown great potential for medical advancements and conservation efforts, it also raises profound ethical, social, and philosophical questions. In this blog, we will explore the science behind cloning, its various applications, and the ethical considerations that surround it. Understanding cloning. Cloning is a process that involves producing genetically identical copies of an organism by using its genetic material. There are three main types of cloning. Gene cloning. This involves the creation of copies of specific genes, which can be used for various research purposes or to develop specific therapeutic treatments. Reproductive cloning. This is the most widely known form of cloning, where an entire organism is replicated. Reproductive cloning has been used to create cloned animals, such as Dolly the sheep in 1996, marking a significant milestone in scientific achievement. Therapeutic cloning, also known as somatic cell nuclear transfer, SCNT, this form of cloning aims to produce embryonic stem cells that can be used to treat diseases and injuries. It has the potential to revolutionize regenerative medicine. Applications of cloning. Medical advancements. Cloning technology holds promise in advancing medicine, particularly in the fields of regenerative medicine and organ transplantation. Therapeutic cloning can create patient-specific embryonic stem cells, reducing the risk of rejection by the immune system and providing a potential solution for various medical conditions. Conservation efforts. In the face of rapid species extinction, cloning offers a ray of hope for endangered animals. Scientists can use cloning to preserve the genetic diversity of species on the brink of extinction, giving conservationists more time to address the root causes of endangerment. Agriculture. Cloning can enhance agricultural practices by replicating desirable traits in livestock and crops. This could lead to increased food production and more resilient and disease-resistant breeds. Ethical considerations. Despite the promising applications of cloning, it also raises several ethical concerns that cannot be ignored. Animal welfare. The process of reproductive cloning often involves a high failure rate, leading to the suffering of numerous animals before a successful clone is created. Ethical questions arise regarding the welfare of these animals. Human cloning. The potential for cloning humans has been a contentious issue since the concept's inception. Ethical debates encompass concerns about the autonomy and identity of cloned individuals, the risk of exploitation, and the slippery slope towards unethical applications. Genetic diversity. Cloning can lead to a reduction in genetic diversity, which is essential for species survival and adaptability to changing environments. A lack of genetic diversity may make a cloned population more vulnerable to diseases and environmental challenges. Unforeseen consequences. Cloning technology is still relatively new, and the long-term effects of cloning on organisms and ecosystems are uncertain. The unforeseen consequences of cloning could have significant ecological implications. Conclusion. Cloning remains a double-edged sword, with the potential for groundbreaking medical advancements and conservation efforts on one side, and profound ethical dilemmas on the other. The discussion surrounding cloning requires a delicate balance between scientific progress and ethical considerations. As we continue to explore the frontiers of biotechnology, it is essential to engage in an open and informed dialogue about the responsible use of cloning technology. Striking the right balance between scientific advancements and ethical boundaries will be critical to harnessing the true potential of cloning for the betterment of humanity and our planet.